The overall goal of this procedure is to quantify vesicular trafficking events in the endocytic pathway of living cells.
This method can help answer key questions in the vesicular trafficking field, such as how mutations or changes in protein expression affect rates and efficiency of trafficking events. The main advantage of this technique is that it allows direct quantification of endocytic trafficking in living cells by measuring the intensity of fluorescently tagged cargo proteins.
After preparing solutions and media according to the text protocol, inoculate 2 to 3 colonies of yeast cells in 5 milliliters of YNB medium, lacking additional nutrients for maintaining plasmid selection. Grow the cultures at 30 degrees Celsius with shaking for 16 to 24 hours. Approximately 3 hours before imaging, measure the OD600 of each overnight culture. Then prepare 5 milliliter dilutions at an OD600 of 0.35 to 0.4 in YNB medium, lacking additional nutrients for plasmid maintenance, and grow at 30 degrees Celsius with shaking for 3 hours. During the incubation, pre-equilibrate the microscope's environmental chamber or heated stage to 30 degrees Celsius. After preparing a Con-A treated 8-well glass-bottomed chamber slide for imaging according to the text protocol, add 200 microliters of YNB medium lacking additional nutrients for plasmid selection to each well. When the cultures are ready, transfer 1 milliliter to a microfuge tube and pellet the cells at 8,000 RPM for 2 minutes. Then remove 975 microliters of the supernatant. Resuspend the cell pellet in the remaining supernatant. Then drop 2.